Alright, let's get started. Hello class, today we're going to be learning about cell differentiation and epigenetics. This will also tie into your epigenetic stations task card activity. To start us off, stem cells. Specifically, we're using stem cells to talk about differentiation, the making of specialized cells. This is basically the whole focus point of stem cells in general. Stem cells, everything stems from them. If you look through the reading and look through things, so the example is, first off, we have the tissue stem cells. So these are stem cells specific for tissue. The example we use will be the blood stem cells. When you're developing, you remember we talked about this, you start off as a little zygote, sperm and egg combined, things like that, and then you start replicating. But at a certain point, you don't just want to be just a wad of the same type of cell, you want to start differentiating. That's what stem cells are for. Blood stem cells can turn into red blood cells, white blood cells, and platelets. All these come from one type of stem cell. Embryonic stem cell, embryonic from the embryo, that's the zygote, the combination of sperm and egg, become everything. Stem cells can either make copies of themselves and make more stem cells, or stem cells can become specialized cells. This human blastocyst, there it is next to the human hair. This is it. This is what we're talking about. Blastocyst, this is about 100 cells. So the sperm and egg have joined, they've started undergoing what process? Mitosis. So they've started undergoing mitosis. Okay, got about 100 cells. At this point, we are at blastocyst. This is when we start seeing some differentiation. This is around the time when stem cells really start kicking it up. So we learned about stem cells when we do an IVF embryos research. So IVF is in vitro fertilization. So in vitro fertilization is essentially where the sperm and egg are mixed outside, like in a test tube, and then the woman is inseminated to cause pregnancy. IVF treatments, sometimes they work the first time, sometimes they work the 10th time. They're not guaranteed, so oftentimes multiple eggs and sperm will be mixed, so set up, you'll have a bunch of embryos ready, and you'll just keep trying until it works. Sometimes it works early on, and there's a lot left over. And sometimes you can freeze them, keep them for later, anything. Some people choose to donate them to research. And through that research, we learned about embryonic stem cells. We're able to begin research on them. Using stem cells, we can test drugs, understand diseases, create new treatments for diseases, and do so much more. But now a common misconception is that people think that's the only place you can get stem cells, and this has been true for over a decade. And since 2007, we've learned that we don't actually need embryos. We can just use regular cells. So in 2007, scientists discovered a way to basically cause regular cells to revert back to their stem cell state. This is a huge boost for science and medicine because just remember, what are stem cells? Stem cells are cells that can become just about anything. So if we reprogram some skin cells, we can get them to turn back into any possible cell. Now, this science isn't there yet, but we are on the way. But the idea is medically, let's say you have a disease that causes your liver to fail and you lose your liver. Now you're living without a liver and that's problematic. What we could do is take some of your skin cells, switch them to stem cells, grow a bunch of them, and then switch them to specialize into liver cells, and we can regrow you a whole new liver. And remember, the process of transplants is problematic because there's rejection. The is technically somebody else's organ, somebody else's DNA, somebody else's cells, and your body can reject it. This would be a new liver grown from your own cells, so there would be zero rejection. This is the future of transplants and regrowing organs and things like that. Specific stem cells, like tissue stem cells, are multipotent. They can become more than one thing. Embryonic stem cells can become everything. So this really wraps up what I wanted you to learn about cell differentiation. There's more to learn than this, but it's just important you understand that these concepts exist. Now, moving on to epigenetics. So we talked about sources of variation in DNA, and you guys have done all this now. Take a look at this huge page. You guys should understand a lot of this. So we learned about how mutation can occur. Specifically, now you know in the S phase where the replication is happening, that's where a lot of mutations will occur. Meiosis, we learned about random assortment and crossing over, things like that. Non-disjunction in chromosomes. Mutagen, mutations, things from the outside mutate your DNA. And now you're ready to learn about the last stage, epigenetics. Epigenetics are reversible modifications of gene expression. This is important from a different way. This is the turning genes on and off using these tags, the methylation and acetylation. Essentially, it's using methyl or acetyl groups to tag it. These are epigenetic markers, so to speak, tag the spots. And that allows certain genes to be turned on and off, which is really important, as we talked about, because you do not want toenails growing in your eyeball. And they are also the part that is influenced by environmental, lifestyle, and dietary factors, aging, and stress. This is where that comes from. This is how the environment can actually impact your genes and how your lifestyle can change the genes you pass down to your offspring. It's not that you're changing the genes themselves, you're changing at what level they're expressed through the alteration of your epigenetics. So epigenetics works on a very simple system. Remember, when RNA was being made, think back to that way back when. It ran along the DNA, reading the DNA as it went, creating the RNA strip that would eventually go out, be read by the ribosome, and create a protein. So epigenetics involves the active wrapping up of DNA to seal it off. It's if you have a tightly coiled bundle section, your epigenetic markers. Here they're called factors. So these epigenetic markers come in and they tag on spots, causing the DNA to bunch up and coil. Now that section of DNA cannot be read, so it will not be expressed. As opposed to over here, you have, because of how the epigenetic markers, the tags, the factors have aligned, it uncoils the DNA, making it much easier to read. This now will be expressed. So this is how, even though you have the DNA for something, it cannot be expressed. It is still coiled up. What we're going to learn about with the activity, which I'll go into in a second, is that some people are more or less coiled than others, and that's what leads to these crazy epigenetic differences. That's the really mind-boggling part. Some person could be partially coiled, so it's partially expressed. Somebody could be significantly uncoiled, so it's overexpressed. These are all possibilities, and that's what epigenetics kind of sits around, because two people could have the same gene, and both are expressing it, but they're not expressing it at the same levels, and that's what epigenetics is. And that brings us to your activity. So we're going to be doing the first one together. We're going to do station 1, lick your rats. Hope you're all excited to lick some rats. You're going to be doing this first station. This first station is actually really important because it teaches you the concept of epigenetics. When you go through, you need to read the short paragraph introduction. It will make a lot more sense if you read it. Then click go on the interactive picture of the rat, and then we're going to complete the interactive 3 times. We're doing it 3 times because we're going to show you how an environmental change can alter your genetics. Specifically your epigenetics. Remember, when we're talking about this, epigenetics and genetics are different. I'm not making or deleting a gene, but I am altering how your gene is expressed. So when we lick our rat pups, we're going to change the speed. The first time we're going to lick the rat pup slowly, then we're going to lick it at a medium speed, and the last time we're going to lick it as fast as we can. If we were doing this in class, I would have you all chant lick, lick, lick as fast as you can, and that'd be more fun. So after that, we'll use the interactive with coiled DNA, and we'll go through the 4 steps. And then we'll be using your graphic organizer to collect evidence to support your claim. Remember, that's the secondary part of this. This is a partial CER. You guys are getting some practice collecting evidence. So your claim you're backing up is there are differences between identical twins because of how genes are expressed and changed by the environment. Make sure you've gone back and watched the twins videos and looked at the twin pictures in the slides before moving on to this point. So you'll be collecting evidence from stage 1. So that's our claim. There are differences in twins because of how they're expressed and changing the environment. So we're going to collect some evidence. So let's begin. Oh no, I need Adobe Flash Player. It's not loading, Mr. Schechter. What do I do? We've talked about this. Remember, you click on it, you get the angry puzzle piece up here, you click the angry puzzle piece, and you click Manage. On the Manage tab, you just flip this switch, exit out of that, refresh the page. This time, when you click it, it'll say Run Flash, and you can click Allow, and the activity will load. These paragraphs talk about how mother rats spend some time licking, grooming, and nursing their pups. Others seem to ignore their pups. Highly nurtured pups tend to be more calm, and pups that receive little nurturing tend to grow up to be anxious. It's important to note we've noticed this in humans too, but we're using rats. First things first, let's lick our rat pups. So right now, it will play you a little explanation and kind of show you what's going on here. So watch through this the first time. I'm just going to start because you can't hear it. So on your marks, we are licking slowly. Ooh, the computer's now critiquing me. So because I failed to lick it significantly, its GR gene has not been activated heavily. The GR gene creates a protein that helps deal with cortisol. Those of you may have heard of it, it's called the stress hormone. Weakened GR genes, GR genes that aren't expressed heavily, means you don't produce a lot of the GR protein, which means you don't deal with cortisol very well. So because I didn't nurture my rat pup early in its life, basically it now will have stress issues. Now we're looking at a medium speed. So now it is more active. It's going to be easier for my pup to relax. Now we're licking it as fast as we can. Lick the rat pup. Lick, 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 lick. Oh, as much licking as possible. Nurture that rat pup. Wow, incredibly attentive rat mother. Go me. Now, because I've done this, the GR gene is incredibly expressed, and now this pup will have an easy time dealing with stress. Now we've shown how the amount of nurturing and environmental stimulation altered how a gene was expressed, and this is the root of epigenetics. Now we can learn about moms, the GR gene itself, how brain cells work, and what happens to an adult pup based on their nurturing. So you can also watch this video to see it happen in real life if you'd like. So remember, you're reading through this, looking through this, and completing the activity with your goal to be to find evidence to back up the claim that the difference between identical twins because genes are expressed and changed by their environment. We just showed how the environmental change, the act of being nurtured at a young age, can change how the GR gene was expressed. That is one piece of evidence you can pull out. You can as well talk about how the impacts it had on the rat throughout its life. So you're going to go through each one of these stations, completing the activities, watching the videos, and filling out your evidence. Remember, evidence needs to be something specific. It is information, scientific data, and facts that will support the claim. It cannot be another claim. For example, if you said that for the rat one, that your evidence was the one that was licked more was happier, or the one that was licked more was less stressed, that's another claim. That's not a piece of evidence. A piece of evidence would be the rat that was nurtured more had a more uncoiled GR gene and thus was expressing the GR gene more strongly. That is a piece of evidence. And because of that evidence, your reasoning could be now that one was less stressed. Remember, that's the key here. A lot of you make the mistakes of stating another claim as your evidence. The evidence is just the raw data, something that happened, and that's fine. Hopefully this helped make stem cells and epigenetics make a little more sense. Hopefully now you can, if you haven't already, you can get started on the epigenetics. Good luck with the activity. Let me know if you have any questions. This has been Checked Biology, Stem Cells, and Epigenetics.